Hi, everybody. So in this tutorial, we are going to explore differences between amino acid sequences among different types of taxa. So this is a common way that molecular biologists will compare evolutionary relationships among organisms using either molecular biology or what we're more familiar with is morphology. But as we know now, we kind of need a little bit of a mix of both of these analyses in order to get a real grand picture of evolutionary relationships among different types of organisms. But the evidence is really strong, especially in something like an amino acid sequence. And if you remember from your upbringing in biology, an amino acid sequence just results from changes in your DNA, or at least your DNA being expressed. And over time, things like mutations can accumulate and cause things to speciate and become different types of species. These mutations lead to different amino acid sequences, and therefore the differences in these sequences for specific types of what are called conserved proteins are going to be able to be compared among organisms to figure out how different they really are from one another and give us a clue as to what their evolutionary relationship is among each other. All right, so that was a lot of an intro. Let's go ahead and get into an example of this and I can explain even more. So first, we're going to work with the conserved protein known as mitochondrial ATPase. It's a type of protein that does a lot of enzymatic reactions in your mitochondria. It's conserved because all organisms will have this protein. There are others examples, but we're going to look at this one, okay? And we're looking at 5 different vertebrate species. Remember, these are your species that have a vertebrae, right? They're ones that you're more familiar with. Okay, cool. We're going to use the table after we've analyzed the amino acid sequence that creates this mitochondrial ATPase from their from biochemical analysis. We're going to look at differences between 5 different taxa. Now, remember taxon, a single taxon is an organism. Okay, it's just a point of interest of evolutionary comparisons. We're going to be looking at E. ferus, D. polylepis, G. gallus, A. foresteri, and E. africanus. Okay, so these are all really interesting organisms. I'm not going to spoil what they are right now because I want you to think about the actual numbers here rather than what the organisms look like or their morphological traits. So right off the bat, there's a couple things we need to think about. Which organisms have the greatest differences? And which organisms have the least differences? Because if we think about this, less difference among your amino acid sequence means that you are more related, you're more similar. And then on the flip side of that, more difference means that you are more different. You probably are not close in relation as opposed to if you had less changes in your amino acid sequence. Just in case you were a little confused as to how to read this table, the very top row here is telling you the 5 different taxa, and the same taxa are going to be mirrored on the column all the way to the left. And that's because they are side-by-side comparisons with one another. This is what we call a pairwise table, a pairwise comparison. In other words, if we go here, E. ferus compared to E. ferus, which is itself, has 0 differences in their amino acid sequence. This makes sense. It's like comparing your own DNA to your own DNA. There should not be a difference, okay? There should not be a difference, all right? So same with D. polylepis, I can never say that word, polylepis, G. gallus, they all have 0s when they're compared to each other. Now let's go to the first with D. polylepis and E. ferus. You can see that that comparison means that there are 19 different amino acid sequences or amino acids in that sequence among these 2 organisms. So that's a pretty high number if you look at all the different numbers in the table here. Now, why am I having you do this? Well, because we are going to need to make a phylogenetic tree that's going to show the differences among these taxa, showing the evolutionary relationships as we are assuming from differences in their amino acid sequences. So don't worry, we're going to work through this together, but remember, go back and point out which ones were the most different and which ones were the most similar. Now, one thing I want to do before we move forward is explain a phylogenetic tree just a little bit in depth. So that way you understand why we can take these pairwise comparisons and put them on a type of genetic map or a map showing you evolutionary relationships. So remember in your phylogenetic trees, it's very, very much like a tree. Now, You might be thinking, what do you mean? Well, if you think about it, a tree, the oldest part of the tree is going to be its trunk, closest to its roots, right? And the newest parts are going to be further away towards the end of its branches. That means that the branch that's stemming off the trunk directly right here, it will be the oldest part. Let me get my spotlight on. It'll be the oldest part of the tree versus this branch that's stemming all the way up here. This will be the newest part of the tree, right? Well, don't forget about that when you are actually thinking about building phylogenetic trees. Knowing that now, and we're looking at our tree and the table of pairwise comparisons of amino acid sequences, let's try and think about this logically. Now, I told you to think about the largest differences versus the smallest differences. Let's start with the smallest. Now, wouldn't it make sense that at these 2 points here, okay, and here, these 2 taxa or 4 taxa will have the littlest differences among themselves? So if that's the case, where are the pairwise comparisons that have the least amount of amino acid differences among each other? Hopefully you pointed out E. africanus and E. ferris, and then A. forsteri and G. gallus, right? Those 2 for sure have the least amount. So they're probably going to be up here in these circles, right? Now, which one has the most differences? So think about it as the one species, the one that would be right here. Which species has the most differences among all other taxa? Pay attention to the highest numbers here. We have D. polylepis with E. ferris with 19. We have D. polylepis with E. africanus with 18. Oh, look, actually, D. polylepis, G. gallus, and A. forsteri, they all have the largest numbers. The common denominator here is definitely D. polylepis, isn't it? So D. polylepis is probably going to be the one that's the oldest divergence because, again, the relationship is the most indifference or the most differences among all the other taxa that are there, okay? So let's go ahead and put D. polylepis on the tree. Okay, so I did a little bit off screen, but I want you to see how we would take the next approach after we put D. polylepis up there. Because again, D. polylepis has the most differences among all the taxa. Now, remember here, these 2 points of divergence, okay, represent a recent divergence. In other words, these 2 taxa on each one are going to have the least amount of differences. So if we go back to our table, that was E. africanus and E. ferris circled here, and it was also, okay, A. forsteri and G. gallus circled here. Okay, now, which one goes where? I have changed the names of the each of these groups as A and B. That way we can kind of just throw them on the tree and see if they make sense. Okay, so I want to go ahead and put group B. Oh, 1 second. Had to scoot things down. I'm going to go ahead and put B. Let's just try this group up here. Okay. And then we will put A right here. Now let's go ahead and look at the table now. Okay, let's look at the table, and I highlighted each of the ones that we are interested in making these comparisons. If you look at D. polylepis versus E. ferris, which is in blue, and E. africanus, which is in blue, that's 19 and 18, versus A. forsteri and G. gallus, which is 15 and 16. Now, if we put A. forsteri and G. gallus on the furthest part of the tree, in other words, the most recent, would it make sense that they have less differences than D. polylepis versus E. ferris and E. africanus? It doesn't really make sense because the group that should end up on this far end of the tree should have the most differences from the opposite end of the tree. Okay, so no, we're not going to put B here. We're actually going to put A. ferris and G. gallus over on this part of the tree because, again, they have, I mean, they still have a lot of differences from D. polylepis, but it's less than E. africanus and E. ferris. So we're going to go ahead and put your A, oops, wrong color. Let's go ahead and make it pink. Okay. A. forsteri, and G. gallus. And then last but not least, we have E. africanus and E. ferus. Now you might be thinking, doesn't matter where I put them on either of these little nodes here, right? Did it matter that I put E. africanus here versus E. africanus or versus here? Well, it doesn't really matter, at least when you're looking at the distal nodes like this. Now, why is that? Well, because you can actually swap this around and rotate it. It doesn't really tell you which one came later versus sooner. What's really important is that you have it on the right node. In other words, they have the same common ancestor. Now, it would matter if I put E. africanus here versus where it is now, because that would assume that there's a different evolutionary relationship there. So be careful. That's not what you want. You know that E. africanus and E. ferus are similar to one another because, again, the one amino acid difference at the very end here. Okay, the very end here. Okay, that was actually supposed to be in blue. My apologies. Let me fix that for you. Okay, so it's fixed. Here is the final product. Now, always sit and think, does this make sense? Again, if you look at the table, the most amino acid differences among taxa are found between D. polylepis and all other taxa. So it makes sense that they are the evolutionarily oldest split right here. Okay, they have the most amount of differences. Okay, and then look at the ones that are the opposite end of the spectrum. D. polylepis had the most differences with your E. africanus and your E. ferus, which is why they are on the distal part of the tree, because they are the most different, right? Okay, so let's kind of explain this a little bit more when we're talking about trying to figure out evolutionary relationships among taxa. Is it better to use morphology, or is it better to use amino acids, or let's say nucleic acids, maybe single nucleotide polymorphisms, right? What is better to show evolutionary differences between things? Well, let me kind of just show you what these organisms actually are if you haven't looked them up already. Well, let's start with the most distal part of the tree, the most recent, which was your E. africanus and your E. ferus. Okay, so these two had one amino acid difference from each other. Now, this kind of makes sense when you're looking at a wild horse versus a donkey, essentially. Kind of makes little sense morphologically. All right, what about the other comparison that only had one difference in amino acids? Well, that was your gallus, right? G. gallus and your A. forsteri. Okay, now they're both birds. Isn't that interesting? Even though one's an emperor penguin and one's I think it's just a wild chicken, but they're very similar to one another. This makes sense that they have only one difference, one change in amino acid. Now, who was on the opposite end, the most evolutionarily oldest? Well, that was actually your D. polylepis, which is the black mamba, which is really interesting. It's a snake, it's a reptile, right? Very different from something like a mammal, like your horses and donkeys versus a avian species, right? Your birds and your penguins. Now, remember that D. polylepis had the most difference with E. africanus and E. ferus, with an 18 to 19 difference between amino acids. And we also had 15 and 16 between A. forsteri and G. gallus. So now, what's the argument here? Well, I think in the end, both molecular biology and morphology are going to be able to help us determine evolutionary relationships. One, even though we did see a very similar comparison or I guess evidence for evolution in a certain direction, in our analysis here with just molecular biology, looking at amino acid sequences, we can actually further support that with morphological data, actually comparing the clades and the different types of taxonomic groups that they actually are found within. For instance, your mammals versus your birds versus your reptiles, right? So both are very, very important, but your molecular biology does help you to see the nitty-gritty behind the scenes of what ends in a specific type of morphology. So lots of fun stuff. Hope you learned a little bit today, and hopefully your tree looks a little something like this.